Hello friends, and welcome to a lecture on quantitative and multifactor inheritance. The main objectives of today's lecture are to understand the patterns of inheritance of quantitative phenotypic traits, to study the influence of genetic and environmental factors on quantitative traits, to define and measure heritability, the unit of inheritance of variation in quantitative traits, to give a precise account of transgressive variations, and to explain the concept of heterosis. Let me first of all begin by highlighting the definition of quantitative genetics. Quantitative genetics is a branch of population genetics that deals with phenotypes that vary continuously in characters such as height or mass, as opposed to discretely identifiable phenotypes and gene products such as eye color in humans and Drosophila, shape of seed and color of flower in pea plant, etc. Moving on, let's now discuss it in detail. The phenotypic traits of different organisms may be of two kinds, which are qualitative and quantitative. The qualitative traits are the classical Mendelian traits of kinds such as form, for example round or wrinkled seeds of pea, structure, for example horned or hornless condition in cattle, pigments, for example black or white coat of guinea pigs, or antigens and antibodies, for example blood group types of man, and so on. Each qualitative trait may be under genetic control of two or many alleles of a single gene with little or no environmental modifications to obscure the gene effects. The organisms possessing qualitative traits have distinct or separate phenotypic classes and are said to exhibit discontinuous variations. The quantitative traits, on the other hand, are economically important measurable phenotypic traits of degree such as height, weight, skin pigmentation, susceptibility to pathological disease, intelligence in man, or amount of flowers, fruits, seeds, milk, meat or egg produced by plants or animals. The quantitative traits are also called metric traits or multifactorial traits. They do not show clear-cut differences between individuals and forms a spectrum of phenotypes which blend imperceptibly from one type to another to cause continuous variations. In contrast to qualitative traits, the quantitative traits may be modified variously by the environmental conditions and are usually governed by many factors or genes, each contributing such a small amount of phenotype that their individual effects cannot be detected by Mendelian methods, but only by statistical methods. Such genes, which are non-allelic and affect the phenotype of a single quantitative trait, are called polygenes or cumulative genes. The inheritance of polygenes or quantitative traits is called quantitative inheritance, multiple factor inheritance, multiple gene inheritance, or polygenic inheritance. Having understood the concept of quantitative genetics, let's now talk about the genetic and environmental factors influencing the quantitative traits. The Danish biologist Wilhelm Johansen was one of the first people to show that variation in a quantitative trait is due to combination of genetic and environmental factors. Johansen studied the weight of seeds from the broad bean Phaseolus vulgaris. Among the plants available to him, seed weight varied from 150 mg to 900 mg. Johansen established lines from individual seeds across this range and maintained each line by self-fertilization for several generations. The seeds from each of these pure lines tend to resemble the seed from which they were found. This ability to establish lines of beans with characteristically different seed weights indicated that some variations in this trait is due to genetic differences. However, Johnson observed that seed weight also varied within each of the pure lines. This residual variation was not likely to be due to genetic differences because each line had been systematically inbred to make it homozygous for its genes. Rather, it must have been due to variation in uncontrolled factors in the environment. Johnson's work, published in 1903 and 1909, therefore led to the realization that phenotypic variation in a quantitative trait has two components, one genetic and the other environmental. Let's now discuss how multiple genes influence quantitative traits. Another genetist, Herman Nilsson L., provided evidence that the genetic components of this variation could involve the contributions of several different genes. Nilsson L. studied color variation in wheat grains. When he crossed a white grain variety with a dark red grain variety, he obtained an F1 with an intermediate red phenotype. Self-fertilization of the F1 produced an F2 with 5 distinct classes, ranging from white to dark red. The number of phenotypic classes expected when there are n diallelic additive loci is 2n plus 1. Thus, the number of F2 classes and the phenotypic ratio that Nilsson L. observed suggested that two independently assorting genes were involved in the determination of grain color. Nilsson L. hypothesized that each gene had two alleles, one causing red grain color and the other white grain color, and the alleles for red grain color contributed to pigment intensity in an additive fashion. Based on this hypothesis, the genotype of the white grain parent could be represented as r, small c, and the genotype of red grain parent could be represented as R, C. The F1 genotype would be R, C, and the F2 would contain an array of genotypes that would differ in the number of pigment contributing alleles present. Each phenotypic class in the F2 would carry a different number of these pigment contributing alleles. The F2 displayed kernels ranged from white to intermediate shades of red in a ratio of 15 red in all shades to 1 white. Among red kernel plants, he could recognize discrete shades. Counting relative number in each class, including white, he found 1:4:6:4:1 phenotypic ratio of plants with dark red, medium, intermediate, light red, and white kernels, which was clearly a modified dihybrid ratio. The explanation is that there are two unlinked loci involved, and the intensity of kernel coloration results from contribution of polygene series of 4 functional alleles, that's contributing alleles. In this and other cases, the effect of contributing alleles on the overall phenotype is additive. Recessive alleles that do not contribute to color are called non-contributing alleles. The white class, for example, would carry none, the intermediate red class would carry 2, and the dark red class would carry 4. Nilsson L.'s work, published in 1909, showed that a complex inheritance pattern could be explained by the segregation and assortment of multiple genes. Let's now talk about variance components of a quantitative trait. The genetic property of a population are determined by the relative magnitudes of the components of variance, that's the genetic and the environmental variance, and thus total variance of a quantitative trait may be mathematically expressed as Vp is equal to Vg plus Ve plus Vge, where Vp is total phenotypic variance of the segregating population, Vg is genetic variance, Ve is environmental variance, and Vg is variance associated with the genetic and environmental interaction. The genetic component of variance may be further partitioned into three components as Vg is equal to Va plus Vd plus Vi, where Va is additive variance, that is variance from additive gene effects. Vd is dominance variance, that is variance from dominance gene action, and Vi is interaction, that is variance between interaction between genes. Additive genetic variance is the variance of breeding values and is primary because of resemblance between relatives. Hence, Va is the primary determinant of the observable genetic properties of the population and of the response of the population to selection. Further, Va is the only component that the researcher can most readily estimate from observations made on the population. The total phenotypic variance may thus be written as Vp is equal to Va plus Vd plus Vi plus Ve plus Vge. Let's now switch over to the concept of heritability, which is defined as the ratio of genetically caused variation to total variation, including both environmental and genetic variation. There are two different estimates of heritability. One is broad sense heritability, that's the proportion of the total phenotypic variance, that's Vp, that's due to genetic factors Vg. It's expressed mathematically as H is equal to Vg divided by Vp. It tends to yield a high value. Some use the symbol H2 instead of H. Another is narrow sense heritability, that's the proportion of the phenotypic variance that's due to additive effects of alleles. Because the additive component of genetic variance determine the response to selection, the narrow sense heritability estimate is more useful to plant breeders than the broad sense estimate. It's estimated as H is equal to Va divided by Vp. Moving on, let's talk about some of the applications of heritability. Heritability estimates are useful for breeding quantitative traits. The major applications of heritability are: number 1, to determine whether a trait would benefit from breeding if, in particular, the narrow sense heritability for a trait is high. It indicates that the use of plant breeding methods will likely be successful in improving the trait of interest. Number 2, to determine the most effective selection strategy to use in a breeding program. Breeding methods that use selection based on phenotype are effective when heritability is high for the trait of interest. Number 3, to predict gain from selection. Response to selection depends on heritability. A high heritability would likely to result in high response to selection to advance the population in the desired direction of change. Now let's move on to another aspect of our lecture, that is transgressive variation or transgressive segregation. In genetics, transgressive variation or transgressive segregation is the formation of extreme phenotypes or transgressive phenotypes observed in segregated hybrid populations compared to phenotypes observed in the parental lines. The appearance of these transgressive phenotypes can be either positive or negative in terms of fitness. If both parents' favorable alleles come together, it will result in a hybrid having higher fitness than the two parents. The hybrid species will show more genetic variation and variation in gene expression than their parents. As a result, the hybrid species will have some traits that are transgressive or extreme in nature. Transgressive segregation can allow a hybrid species to populate different environments or niches in which the parent species do not reside, or compete in the existing environment with the parental species. Now let's come to the causes of transgressive segregation. First are the genetic causes. There are many causes for transgressive segregation in hybrids. One cause can be due to recombination of additive alleles. Recombination results in new pairs of alleles at two or more loci. These different pairs of alleles can give rise to new phenotypes since gene expression has been changed at these loci. Another cause can be elevated mutation rate. When mutation rates are high, it's more probable that a mutation will occur and cause an extreme phenotypic change. Reduced developmental stability is another cause for transgressive segregation. Developmental stability refers to the capability of a genotype to go through a constant development of a phenotype in a certain environmental setting. If there is a disturbance due to genetic or environmental factors, the genotype will be more sensitive to phenotypic changes. Another cause arises from the interaction between two alleles of two different genes, also known as epistatic effect. Epistasis is the event when one allele at a locus prevents an allele at another locus to express its product, as if it is masking its effect. Therefore, epistasis can be related to gene overdominance caused by heterozygosity at specific loci. What this means is that heterozygote, when compared to the homozygote, is better adapted and therefore show more transgressive or extreme phenotypes. All of these causes led to the appearance of these extreme phenotypes and creates a hybrid species that will deviate away from the parental species niche and eventually create an individual hybrid species. Second are the environmental causes. Other than the genetic factors solely causing transgressive segregation, environmental factors can cause genetic factors to take place. Environmental factors that cause transgressive segregation can be influenced by human activity and climate change. Both human activity and climate change have the capability to force species of a specific genome to interact with other species with different genomes. For example, if a bridge is built that connects two isolated areas to one another, a gene flow door would open. This open door will increase the interactions between different species with different genomes, which can create hybrid species that can potentially show transgressive phenotypes. Human activity can open the gene flow door by pursuing harmful actions such as cutting down forests and pollution. Climate change, on the other hand, can open the gene flow door by breaking climate and environmental barriers that were present before. This convergence between species can give rise to hybrid species that will have more phenotypic variation when compared to the parent species. This increase in phenotypic variation has the potential for transgressive segregation to occur. Let's now understand what is heterosis. Heterosis is a measure of the superior performance of the crossbred relative to average of the purebreds involved in the cross. The probable cause of most heterosis is due to combining genes from different breeds. concealing the effect of inferior genes. Heterosis may result in the crossbred being better than either parent breed or simply better than the average of the two. Having understood the concept of heterosis, let's discuss about genetic basis of heterosis. The genetic basis of heterosis is the opposite of the origin of inbreeding depression. Inbreeding tends to cause more gene pairs in an individual to be homozygous, that is, the offspring receives an identical gene from each parent. In contrast, crossbreeding tends to cause more gene pairs to be heterozygous, that is, the offspring receives different genes from its parent. This arises from the fact that different breeds tend to have high frequencies of different genes. Friends, that's all about today's program. Hope you have understood and enjoyed. Thank you and goodbye.